Sato Company.
How does human life begin? We all start as one tiny fertilized egg. How do we evolve into such complex and sophisticated organisms? This has been one of mankind's biggest mysteries, one that scientists around the world have attempted to unravel. Our understanding of this process has grown rapidly in recent years. Research into embryonic stem cells and iPS cells used in regenerative medicine has played an important role in solving this mystery. Technological advances are finally opening the doors of understanding. Researchers have found incredible mechanisms deep within the fertilized egg. The key players are the messaging molecules, which are used by cells to communicate with each other. Scientists now know that messaging molecules are released at different stages, so that our organs can start forming on their own.
I think the surprise for me was the amazing degree to which the cells sort of know what to do. They're constantly communicating with one another and instructing them what to become.
This exchange of messages also causes amazing transformations inside the mother's body.
So part of the placental cells seek out the mother's blood vessels and invade the walls.
The baby uses astonishing means to ensure its survival. The veil is finally being lifted on something that was once unknown, the incredible drama of life before birth. Join us to witness this fascinating story.
What does it take to live a full and healthy life? A vast information network exists within the human body, where our organs constantly exchange information with one another. Now, let's listen in to the conversations that sustain our lives.
December. We decide to trace the journey of life from its very beginning. Megumi and Atsushi Matsunaga have been married for 3 years. Megumi had undergone surgery for fibroids, and the couple were having trouble conceiving a child. Gynecological tests raised additional concerns.
卵子の数が極端に少ないっていう話で、3年のうちに子供をっていうことだったので。
With their odds of pregnancy decreasing, the Matsunagas made a decision.
思い切って体外受精。
The procedure would involve fertilizing the mother's eggs with the father's sperm in the laboratory. With the help of the latest medical technology, the moment of conception could be captured in vivid detail.
人の命がどんな風に誕生していってるかっていうことがより克明に分かってくるんじゃないか。
Here you can see the sperm heading toward the egg. It enters the egg. But it's not over yet. The two round spots are the pronuclei. One contains the genes from the mother and the other from the father. The genes packed in the head of the sperm swell and emerge. And then the contents of the two pronuclei suddenly disappear. It is at this very moment that new life begins. This miraculous moment was captured for the first time using a special microscopy technique. When the two pronuclei disappear, every single individual chromosome is clearly visible. They are now gathering together. And the genes of the mother and father merge to create a new life. 2 hours later, the egg begins the cell division process. Over 5 days, the number of cells increases to about 100. Then it's time for the embryo to be implanted in the uterus.
最初これを渡されて、この受精卵を戻しましたっていうので。
However, a key question remains. Will the embryo successfully implant itself? If not, there will be no pregnancy. Scientists have discovered that a certain messaging molecule initiates a dramatic change inside the mother's body. What happens exactly? The first person in the world to record this crucial phenomenon was Professor Ali Brivanlu.
We had never seen this before because, again, it's impossible to have access to this biological material, especially knowing that women don't know even that they're pregnant.
Let's observe the embryo after implantation in the simulated environment of a special cell culture dish. It starts to release a substance shown in yellow. Over a period of days, this substance keeps increasing. It's called HCG, and it contains the first message sent from the embryo to the mother. HCG plays an important role in making sure that the embryo takes root in the uterus. Here, the embryo has attached itself to the uterine wall. However, left on its own, it will be shed from the mother's body when her menstrual period arrives. So the embryonic cells send out a message molecule. This message makes its way throughout the mother's body. In response, her body blocks menstruation, and it thickens the uterine wall. The embryo becomes firmly embedded.
These cells are those who are going to be talking to the maternal cells to establish all the inductive interactions and communications that are required.
HCG is already used in pregnancy tests.
C'est une patiente qui est vraiment un résultat bien positif, qui est certainement de 2 à 3 semaines enceinte. Elle sera heureuse de pouvoir l'apprendre.
For a woman trying to get pregnant, the presence of HCG in the blood is proof that she has taken the first step toward motherhood.
If there isn't any HCG detectable, then it's quite easy, she's not pregnant.
3 weeks since the embryo was implanted in Megumi's body. Megumi is about to receive the results of her blood test. The chance of becoming pregnant in a single round of vitro-fertilization is about 30%. Megumi's blood contained a substantial amount of HCG, sending a strong message that she is indeed pregnant.
目見えますか？ちっちゃいやつ。
すごい。The embryo has been firmly established in the thick, soft tissue of the uterine wall.
無事でよかったです。安心しました、少しだけ。はい。
Congratulations to the Matsunagas. However, it is still the early stages of this journey. There will be many things that must be done. The human body is made up of many types of cells. They come in many different sizes and shapes. Some form the stomach, others the intestines, and all these cells work together to form the basis of life. All these different cells originate from one single fertilized egg. How is this possible? This was one of the great mysteries of embryonic development. But scientists have discovered that the answer is also found in messages exchanged among cells. Scientists have been trying to solve the mystery of birth for centuries. Recent research has led to major progress in unraveling this mystery. Kenneth Chen, a leading international expert in regenerative medicine, points out that one discovery led to huge advances in the embryonic development.
The major advance, of course, was the discovery not only of human embryonic stem cells, but human iPS cells. Of course, Dr. Yamanaka in Japan was the pioneer in this area, and this has had a transformative effect in our field.
Both embryonic stem cells and iPS cells are similar to fertilized eggs after they start to divide. If we grow these cells in the lab, we can observe how cells divide and turn into various organs. In Professor Chen's lab, human embryonic stem cells are grown and developed into cells of one particular organ. A special signal is added to the embryonic stem cells. The messaging molecule is called Wnt. Wnt molecules are present in large quantities in dividing cells. 1 week later, some of the cells have begun to move. 2 days later,
この2つなんですけどね、拍動しているのは肉眼でもわかるぐらい。
The entire sheet of cells has started to ripple like a wave. This is the very beginning of a human heart.
You can take an embryonic stem cell that can become any cell and then instruct it to specifically, almost entirely become human beating ventricular muscle. This is an exciting event. And those instructions generally are going to be, I believe, signals that come and tell the cell to go in one direction or another.
Scientists can now capture extremely detailed images of the embryo. The embryo continues to divide. In a very early stage, before any organs have formed, the cells start to change. Wnt signaling begins.
It is here where the signaling, they're first exposed to different signaling levels that will tell them to go on to form different cell types.
The heart is the first organ to emerge. And once the heart is formed, this triggers yet another dramatic change. What happens? We were able to capture this rare moment on camera. Dr. Takanori Takebe is attracting worldwide attention for his research that induces iPS cells to turn into the cells of various organs. On this day, his team is monitoring heart cells created from human iPS cells.
面白い。これですね。この拍動してない子たちはもしかしたら肝臓のこう初期的な構造物が出来上がりつつあるのかもしれない。
Right next to the beating cells of the heart, what's believed to be liver cells have already appeared. Takebe's research revealed that heart cells stimulated the neighboring cells to start forming the liver. Pioneering international research has unveiled the process by which embryonic cells generate organs. The fertilized egg starts as a single cell and divides inside the uterus. Once it reaches a certain number of cells, a messaging molecule goes to work. The cells that receive the message start to turn into heart cells. Then, the newly formed heart sends out the next message. This message is received by nearby cells, and the liver is formed. Gradually, as more messages are transmitted in complicated and interrelated ways, organs such as the lungs and pancreas are created in sequence.
人の体の中では単一の臓器だけ作るということって全く起きませんので、いろいろな臓器でお互いの臓器近くにあるもの、遠くにあるもの、みんな会話をしていると思います。
The same exchange of messages has started inside Megumi as well. On the day when implantation of the embryo was confirmed, it was a mere 3 millimeters in length.
これ赤ちゃんの心臓の動きですね。
Its first organ, the heart, has started to beat. This is the starting line. Inside, a series of messaging molecules are being sent out. Here we see hands and feet. That's also due to the messaging molecules. About 2 months later, if we look near the heart, Just underneath is the liver. The lungs and intestines are also in place. The black spot is the stomach, where amniotic fluid has pooled. Most of the organs can be confirmed, and the basic elements of the human body have formed.
順調です。
はい。だいぶいっぱいになってきましたね。
The organs continue to form thanks to the work of the messaging molecules. But more mysteries remain. Let's take a look at the internal structure of the organs. This is the inside of an adult lung. The bronchial tubes have a complex structure, with each tube splitting into smaller branches more than 20 times. The interior of the tubes also has a highly complex structure. The interior is completely covered by cells. And if you look closely, you can see more than one type. Between the furry cells, there are other cells at regular intervals that secrete a substance. This structure is essential for removing dust and pathogens. Every single one of our organs are filled with complex microscopic structures like this. So how are the internal structures of organs formed? Professor Takashi Miura is using computer simulation to study the mechanism which make bronchial tubes grow. Scientists believed that bronchial tubes grew in response to a message sent by surrounding cells saying, "Grow further." However, a very complex program would be needed to create the intricate branching structure with the messages from neighboring cells alone. So Professor Miura included a different message sent from the bronchial cells themselves into his calculation. The message is, "I don't want to grow." When Professor Miura tried a simulation that involved the messages from the cell types interacting with each other, he found that even an extremely simple program could recreate the structure of the bronchial tubes.
お互いにこうなんかシグナル出し合って形のルールができてくると勝手にこういう綺麗な枝分かれ構造というのができてきます。事情構造が複雑だからといって作るメカニズムも複雑である必要はあまりなくて非常に単純な2つの領域の間の相互作用だけでもこういうような構造が作ることができます。
And yet, another messaging molecule gets involved in the development of the two different types of cells appearing on the interior of the tubes. This was discovered by Mitsuru Morimoto of Riken.
この2種類の細胞はもともとは同じ性質を持った幹細胞だったんです。ところが分化する瞬間にこの細胞が隣にシグナルを出します。そうすると隣の細胞は緑の細胞ではない細胞にならなきゃいけないというシグナルが入ります。でこのシグナルが繰り返し起こることによって細胞は緑赤緑赤という入れ子のパターンになっていきます。
By using a special message that can only be communicated to the next cell, a structure in which cells line up in an alternating pattern can be easily created.
お互いに話をして、みんなが同じものにならないで多様なものを作ると多様なものを作ることによって、組織は臓器は機能を獲得することができるようになります。
The messaging molecules not only determine the location and shape of organs, they also determine the way the cells align themselves. The human body is indeed created by messages. At the same time that it forms organs, the baby growing inside Megumi must also undertake another important task.
最初のこの辺は本当に虫みたいだよね。この辺からちょっとずつ宇宙人になってきたかな。
The baby has to grow bigger. Let's imagine that the fertilized egg was the size of an apple with a 10-centimeter diameter. How big do you think the newborn baby would be? Amazingly, it would be 500 meters tall. That is how much a baby grows in just 40 weeks. This astronomical growth is supported by a special organ. It's the placenta. The baby's umbilical cord connects with the mother's uterus here. It's another vital organ that's created from the fertilized egg through division and is therefore part of the baby. The placenta provides the baby with the oxygen and nutrients it needs to grow. Let's take a look to see what happens inside. See the tree-like structures? We'll call them baby trees. Blood vessels from the baby run throughout the inside of the trees. This is the frontier of the baby's body. And up above is the wall of the mother's uterus. We can see the openings where the mother's blood vessels end. Maternal blood filled with oxygen and nutrients comes showering down from them. The baby trees absorb nutrients from the mother's blood. Oxygen is also absorbed through the branches. As soon as the red blood cells hit the branches, oxygen is passed on. But the maternal blood is also sending an important message. Grow more. In response, the baby trees spread out further. This is how the increased oxygen and nutrients necessary for the baby to continue growing are delivered. Scientists are continuing to discover more and more messaging molecules that are working inside the placenta. Detailed genetic analyses are now helping us to create a complete picture of the messaging molecules being exchanged in the placenta.
So what we see is interesting is a lot of crosstalk going from both sides. So the maternal and fetal side communicate a lot from the mom to the fetus, as well as, of course, a lot of signals also coming back on the other side, from the fetus to the maternal side.
Nearly 100 messaging molecules were found. What messages do they transmit? Researchers are still working to decipher their functions. One of them has the power to dramatically influence the baby's growth. It triggers a remarkable change inside the mother's uterus. Megumi is now entering her 4th month of pregnancy.
体調いかがですか？つりがちょっとひどいですけど、食べたものは戻しちゃうのと、あと水分もなかなか取れなかったり。
She still has about a month before her condition stabilizes. During this time, mothers worry if the baby is growing as it should. It is only 10 centimeters long, and inside the head,
ここがね、大脳なんです。この薄いのは。ちょっと厚くなってきてるのがわかりますかね。
the cerebrum is the part that looks like a thin casing around the brain. There is a big gap between it and the core of the brain. Not only the brain, but other organs also still need to grow quite a bit. Around this time, something extraordinary happens inside the uterus. To continue the baby's growth, the mother's body literally takes the pain. Professor Susan Fisher was the one who shed the light on this process. She focused on the opening of the maternal blood vessels, from which oxygen and nutrients come showering down. These images, taken with an electron microscope, are vessels during early pregnancy. At a glance, they don't look much different. But when we show them at the same scale of magnification, the difference is clear. The opening on the right is 10 times bigger. Research on the cells in these vessels has uncovered a surprising phenomenon. If we enlarge the image of the opening, we can see that some cells, in yellow, are slightly different from those nearby. Let's look more closely. They look like they have very fine villi on the surface. These cells, in fact, come from the baby. Lots and lots of cells from the baby trees are gathering on the wall of the mother's uterus.
So part of the placental cells have specialized molecular homing devices to be able to seek out the mother's blood vessels and invade the walls.
Let's witness the dramatic events that take place in the placenta to spur the baby's growth. This is the inside of a placenta in the 4th month of pregnancy. The baby trees have grown quite large. However, the tips of the branches have turned slightly black. As the baby grows, the mother's blood can no longer supply all of its needs. Oxygen and nutrients do not reach every branch. If something is not done quickly, the baby will stop growing. Now, the baby trees have started to send something out. It is the messaging molecule PGF, which contains a message that's essential for continued growth. Once the mother's uterus receives the message, the mother expands her blood vessels to allow a larger flow of blood. But that's not all. The branches continue to stretch until they reach the uterus. The cells of the baby trees invade the uterine wall. They have penetrated into the opening and into the mother's blood vessels. They break through the wall of the blood vessels and squeeze their way in. And then the wall of the mother's blood vessels disintegrates, and the opening expands even wider. A great gush of blood flows over the baby trees. Part of the mother's body has been literally torn open. This aggressive event is necessary to provide the nurturing shower of life-giving blood. The flood of oxygen and nutrients received through the baby trees is sent to the baby through the umbilical cord. This is how the baby is able to continue growing. This significant change is also occurring inside Megumi's body. But Megumi is worried about something. Before becoming pregnant, she had surgery for uterine fibroids.
多発性の筋腫だったので十何個あったんです。これは子宮全摘出だなっていう話をそこでされまして、子供はもうダメかも。その時は辛かったですね、私も。
In the end, Megumi managed to avoid a hysterectomy, but a large amount of her uterine lining had to be removed, leaving the uterine wall much thinner. Megumi is worried whether she will be able to continue to supply the large amount of blood her baby requires. The baby is due in a month. The doctor is checking the blood flow to the placenta.
これ胎盤ですよね。血液がしっかり流れ、酸素と栄養が運ばれると。
Megumi's uterus is providing an ample supply of blood. The baby is receiving it and has grown considerably. The area which was empty before is now full of brain cells.
もういつ生まれても大丈夫なように準備ができてる。
And finally, today is the day.
よし、頑張らないと大丈夫。
Megumi's fibroid operation created another problem. It left her uterus at risk of rupturing during delivery, so she's having a cesarean. 1 hour into the procedure.
お食べください。よいしょ。はい、おめでとう。おめでとうございます。お食べください。
It's a healthy baby girl.
松永さんおめでとうございます。2164グラム。触っていいですよ。
Through the exchange of messages between the mother and fetus, a single fertilized egg has become a baby. This is how we were all created. An epic story lies behind the birth of every baby. The phenomenon in which the fetal cells break through the uterine wall occurs only in humans and certain other closely related mammals. This is thought to be because we develop very large brains as fetuses. We rely on our mothers to provide the oxygen and nutrients to grow our larger brains. Another reason to thank our mothers. The quest to solve the mystery of birth has led to a revolution in the way we save lives. Landry is 12 years old. She has impaired function in multiple organs, including her bladder, intestines, and gallbladder. This may have been caused by disruptions in the messaging molecules before she was born.
By here, you get an IV every day where you get nutrition. Can you show them? So what's it like to get TPN?
Well, it's kind of weird, but it helps. So.
How long are you on TPN a day?
She has had repeated operations, but recently the condition of her liver has deteriorated.
So what do you think about that?
Takanori Takebe, the scientist researching the process of organ development, has become involved in her treatment. Takebe is developing a treatment using iPS cells to create a mini liver to be implanted in the patient's body. At the frontier of regenerative science, scientists around the world are tackling similar approaches. But even if he succeeds in generating liver cells, that doesn't mean he can create a functioning organ. The liver is a complex organ. This image of living liver cells was captured by an 8K microscope. The cells in blue are liver cells, and the red parts around them are capillaries. For the liver to function properly, it needs this complex tissue structure. It was long thought to be impossible to create such a complex structure artificially. So Takebe is mimicking the process that takes place inside the uterus. He uses iPS cells that are turning into liver cells, then adds blood vessel cells and other support cells that accompany the process. This creates conditions that are similar to those when a liver is formed inside a fetus. Then the cells are cultured in an environment similar to that of the mother's uterus. Takebe hopes that the cells will send out messaging molecules and interact to form the complicated liver structure on their own. After repeated experiments, the dispersed cells start coming together on their own.
きれいきれい。やっぱり締まってますよ血管。そうですね。違いが出てる。出てる出てる。
The result was a tiny liver measuring just a few millimeters and filled with a network of blood vessels. But will it function properly inside a living body? The small livers were implanted in mice with impaired liver function. The 30-day survival rate of these mice increased significantly. a miniature liver which actually functions in the body had been successfully created for the first time in the world. Takebe wants to apply his research now to helping people. So in 2017, he launched a lab to produce these mini livers in quantity. He hopes to start clinical trials in a few years on children with severe liver diseases.
やはりああいう子に生かすために僕ら研究やってきているので、ミニ肝臓を使ってあの子の病気を治すってことができたらいいな。非常に近い将来ですね、実際役立つ技術として使ってもらえるんじゃないかと思っています。
Research into creating artificial organs is accelerating around the world. These functional mini organs are called organoids.
So these are stomach organoids, and these are intestinal organoids.
Stomach organoids release stomach acids. And intestinal organoids move food and expel waste using peristalsis. Kidneys have the most complicated structure in the human body, but parts of their structure have been successfully created too.
腎臓がね、目の前でこうできていく様子をできているよねっていうのが分かった時の感動っていうのはね、やっぱこれはね、涙なくしてはね語れないぐらいね嬉しい話なんですよ。まあこれから劇的にまたどんどん進むんじゃないですかねと思いますね。はい。
These organoid technology gives us a window to understand each of these different organs, and ultimately it will be important to synthesize all of this together to get a more complete picture of the mysteries of the human body.
Every day around the world, new life continues to emerge. Every single birth is a miracle. We all started our journey from a single fertilized egg. That's how your story began. One by one, messaging molecules were released and our organs were formed. Then, when you sent out messages to allow further growth in the womb, your mother's body responded, continuing to provide you with her riches. That is what made you the highly functioning and complex human being you are today. The Matsunaga's baby is now 5 months old. Her weight has tripled since birth. How were we born? You now know some of the answers to this question. This was the story of the beginnings of life. A true story for everyone.